Hello students, welcome to EPG Pathshala. I am Divas Pradhan, scientist in Dairy Microbiology Division, National Dairy Research Institute, Karnal, Haryana. Today I am going to talk about food science and omics era, genomics, proteomics, nutrigenomics from the paper Advances in Food Science and Technology. And introducing let food be your medicine and medicine be your food was long quoted by Hippocrates in 400 BC. The advancement of technology has now changed the way with which we look at the food. Food is now considered today not only a source of energy but also an affordable way to prevent future diseases. The interaction of foods and individual food constituents with the biological systems can now be defined on a very molecular basis with the advantage of omics technologies. In the past, nutrition and food research mainly focused almost exclusively on the metabolites, which involved either measuring the concentration of few individual intermediates or markers, whereas the new era of omics technology enable insights in virtually every step in the processing of the biological information, from the DNA to the RNA to proteins and also to the metabolic functions of the individual proteins. This new area of science, which is also referred to as nutrigenomics or nutrigenetics, respectively, will increase our fundamental knowledge of the interaction between different life processes and our diet or specific components thereof, which may in time lead to the development of either novel functional foods to improve the health status of the general population or to the personalized diet to prevent the onset of nutrition-related disorders in genetically predisposed individuals. Nutrigenomics is a branch of nutritional genomics, and it is the study of the effects of foods and the food constituents on gene expression. This means that nutrigenomics is a research that focuses on identifying and understanding the molecular level interaction between the nutrients and other dietary bioactives with the genome. The broad objective of this module is introducing to the concept of omics science. What are the various components of the omics science that interplay in between themselves to bring about the final change, and again the application of the whole concept in nutritional science. Omics science. The figure here typically shows the flow of information or the central dogma for the flow of information. The genetic material, the study of the genetic material is also known as genomics. The mRNA that comes from transcription of the gene is the study of the whole pool of mRNA is called the transcriptomics. mRNA after translation comes to protein, and the study of the whole protein is also known as a proteomics. Proteins lead to formation of different metabolites, and the study of the whole metabolite is also known as metabolomics. The metabolites, after different interplay and at the different condition, it leads to the expression of the phenotype. Omics, the execution of a human genome project, brought forth a wealth of information about the structure of the genome, which can now be used to study how the interplay between our genes and factors from the environment like the nutrition relate to a state of health or disease. To enable such studies, novel technologies have been designed, in particular to monitor the activity of multiple genes simultaneously as well as on the level of the RNA by transcriptomics or on the level of the proteins by proteomics. Hence, omic technology, as it is popularly known as, is a collection of technologies that aims at characterization and quantifications of pools of biological molecules at different levels that translate into the structure, the function, and the dynamics of the organisms or the organisms as a whole. It mainly surpasses technologies, the popular technologies such as genomics, the transcriptomics, the proteomics, the metabolics, and other as well. Nutrients and non-nutrients components of foods, diets, and lifestyle, including physical activity, can affect every step in the flow of genetic information from gene expression to the protein synthesis, and thereby ultimately alter the metabolic function in complex ways. These technologies can be used for identifying the effect or mode of action of foods or particular ingredients, and hence are finally leading to a science-driven development of foods with a very defined biofunctionality. Genomics. Genomics is a discipline in genetics that typically deals with recombinant DNA, DNA sequencing methods, and bioinformatics to sequence, assembly, and analyze the function and structure of the genomes, that is the complete set of DNA within a single cell of an organism. The omic study starts with genomics. The genomic analysis of an organism typically involves three major broad components. That is mainly, the first one is a sequencing of DNA. DNA sequencing was first popularized by a technique developed by Frederick Sanger in 1977 that was based on principles of chain termination reaction. However, with the arrival of advanced sequencing platforms such as spiral sequencing, shotgun sequencing, ion torrent sequencing, etc., DNA sequencing has become much faster and cost effective. The assembly of that sequence to create a representation of the original chromosome is the second part of genomics. Sequence assembly refers to aligning and merging fragments of a much longer DNA sequence in order to reconstruct the original sequence. This is needed as current DNA sequencing technology cannot read the whole genomes as a continuous sequence, but rather read small pieces of between 20 and 1000 bases depending on the technology being used. The third part of the technology is the annotation part, and again the analysis of that representation. It is the process of attaching biological information to sequences that consists of identifying portions of the genome that do not code for proteins also. It also includes the identifying elements on the genome that is also called by the process gene prediction and attaching biological information to these elements. Sequences from different organisms can also be compared for homology to determine the already known functions. Genomics. To better understand the functionality of vast amount of data generated through various genome sequencing projects, concepts such as functional genomics and structural genomics have emerged. Functional genomics, it tries to understand the relationship between an organism's genome and its phenotype. It includes a function-related aspects of the genome itself, such as mutation and polymorphism, as well as the measurement of molecular activities. Now, structural genome, on the other hand, it seeks to describe the three-dimensional structure of every protein coded by a given genome. This genome-based approach allows for a very high throughput method of structure determination by a combination of experimental and modeling approaches. The second part of omics study is transcriptomics. Transcriptomics is the study of the complete set of RNAs, that is also called the transcriptome, encoded by the genome of a specific cell or organism at a specific time or under a specific set of conditions. It is also referred to as expression profiling because it examines the expression level of mRNAs in a given population under defined conditions. The most popular tool for RNA profiling is the DNA microarray or DNA microchip array, which is generated by spotting a piece of each target gene on a solid carrier. The application of DNA microarrays for RNA profiling is based on the principles of hybridization, the selective binding of each mRNA as complementary DNA or cDNA to its complementary gene fragment. After washing, the microarray is scanned to detect the fluorescent signals. If a gene does not change its mRNA production due to the treatment, similar amounts of Cy3 and Cy5 cDNA will be present on the spot of that gene, which should light up in yellow. Now, green and red are the colors coming from the spots with an abundance of cDNA either from the control and treated cells respectively. Based on fluorescent staining intensities, the observed changes in mRNA production of each active gene can be quantified using various softwares, and the expression of each gene can be profiled accordingly. In preparing for the analysis, the mRNA is first converted into cDNA by reverse transcription process, and in the process a fluorescent dye is also attached to it. Commonly used fluorescent dyes such as Cy3 that gives green color and Cy5 that gives red fluorescent dye are employed, and lightning is caused by the irradiation by UV light irradiation that occurs at different wavelengths. Given in the figure below, it is seen that the complementary DNA from control cells before treatment can be labeled with Cy3 as against the complementary DNA after treatment, which has been labeled with Cy5. To visualize changes in the mRNA production, both labeled complementary DNA samples are mixed in a 1:1 ratio and hybridized to the microarray, where each cDNA will bind to its spotted DNA of its own gene. This figure here shows the process of transcriptome analysis or gene expression that has been mentioned before. In this case, there are two stages, that is the control cells and treated cells. The RNA is isolated by the traditional method. The isolated RNA is then converted into cDNA by reverse transcription, and in that process the dye, that is Cy3 or Cy5, is stacked depending on whether it is the control cells or the treated cells. It is then mixed, as already mentioned, at a defined ratio of 1:1 and then put into the plates where it has been impregnated with the DNA. The hybridization process takes place, and upon visualization using different softwares, we can see the spots lighting up, and which can be accordingly quantified to check the expression of each and every target gene accordingly. The third stage of omics science is proteomics. Proteomics is the large-scale study of proteins, particularly their structures and functions in an organism or a cell. It is known that proteins are vital parts of living organisms, hence its systematic study would provide a comprehensive view of the overall structure, function, and regulation of the biological systems. The proteome is the entire set of proteins that is produced and modified by an organism or system upon a certain response. Whole cell protein analysis begin with the isolation of whole proteins from a biological sample or a cell extract. Protein isolation is mainly done by chemical methods that mainly uses the precipitation or differential solubility as the main principle. The protein extracts or the peptides that is the products of proteome digestion, are then fractionated by methods such as the two-dimensional polyacrylamide gel electrophoresis, that is also known as 2D page, or liquid chromatography. In 2D page, individual proteins are separated in polyacrylamide gel based on isoelectric point, that is of course the first dimension, and molecular weight, that is the second dimension. After staining, proteins appear on the gels as individual spots, of which the staining intensity is used to estimate the relative amount of a protein in the sample. However, looking at spots does not actually reveal the identity of the proteins of interest. For this, mass spectrometry in combination with the knowledge of the sequence genome is used. This figure depicts the analysis of proteins from a cell or organism. The cell is cultured under specific conditions and protein is isolated depending on different principles used. The protein extracts is run in 2D electrophoresis, and depending upon the two different dimensions, proteins are visualized as individual spots in the 2D gel. This is again applied into a mass spectrometer, which depending on the M/Z ratio that has already been mentioned before, it gives a different peaks, and depending on the peptide profiling, mass determination of intact proteins can be determined, and which finally leads to the protein identification of the target protein. Mass spectrometers operate by converting the analyte molecules to a charged ionized state with subsequent analysis of the ions and any fragment ions that are produced during the ionization process on the basis of their mass to charge ratio, that is also given by M/Z ratio. A protein is first treated with a proteolytic enzyme popularly used is trypsin, which cleaves the amino acid chain behind every lysine and arginine. Since every protein has its own specific gene-coded order of amino acids, tryptic digestion results in a protein-specific mixture of fragments. A protein-specific mass spectrum is generated from a mixture showing an M/Z peak for each fragment, which is referred to a mass peptide fingerprint. Here again we combine the knowledge from the genome. It comes in because the sequence of most genes, at least for the humans, is known. The coding part of each gene can be translated in silico into an amino acid chain, which can then be cleaved in silico by trypsin. Calculating the exact mass of each tryptic fragment gives a theoretical peptide fingerprint for every gene in the genome. By comparing the experimental peptide fragment with all the theoretical fingerprints, a match will reveal the identity of the protein. Metabolomics. Although metabolites are in fact not the product of a gene, their existence and again the concentration depends on the activity of multiple activity of genes. Therefore, metabolomics, that is the large scale analysis of multiple metabolite concentration under changing nutritional conditions, is also regarded as being part of nutrigenomics. Metabolomics may be especially suited to assess exposure to nutrients or for measuring compliancy during a dietary intervention in specific diseases, or again the determining the bioavailability of specific nutrients. Different technologies are applied, of course, for the profiling of different classes of metabolites like lipids, carbohydrates, and amino acids. However, the most important technique that is now very much popular for metabolic applications in human biofluids are gas chromatography, that is also called as GC, or liquid chromatography, LC, that is again coupled to mass spectrometry, MS, or nuclear magnetic response spectroscopy, that is also called as NMR, to obtain the most valuable information from the metabolome a combination of different techniques appears necessary. Because each technique covers a different mass and concentration ranges, so again we have application of different forms in different forms. For example, GCMS is mainly used to identify and quantify the low molecular weight metabolites with masses of up to 1000 daltons. In nutrition science, GCMS has been used to determine the fate of specific food components or metabolites. LCMS seems particularly better suited for the analysis of labile or low to intermediate molecular weight compounds. It is the most global of the techniques described here and can be again used to determine a raw metabolite profile in addition to identifying and quantifying specific compounds. Absolute quantification, however, requires reliable internal standards, and better results for identification can again be obtained using tandem MS, LCMS or MS. Application in food science. Application of omics science in food science can have diverse applications. However, it has been divided into two main deliverables. This either result in the identification or selection of specific biomolecules controlling or representative of a biological process, or they use the complete biomolecule set to assess the response or quality of the transcriptome, proteome, or metabolome. The first application of omics science in food science can be used for screening for novel bioactive food components. The availability of rapid screening methods for detection of bioactivity will highly speed up the process of identification of new bioactives for functional foods. Using the omic approach, such as gene expression or proteome profiling, it would be possible to rapidly screen a number of food components for specific bioactivity. The second use of omics science in food science may be used for the assessment of safety of food components. Apart from bioactivity, it is of tremendous importance to evaluate food components for safety, since adverse side effects are not tolerated for foods. Similar to bioactivity assessment, using omic technology, these food components can be checked for specific safety-related gene or protein or metabolite patterns that shows any divergence from the safety, so that it can be safely used in functional foods without any diverse effects. The third use may be detection and control of food spoilage or pathogenic microorganisms. Omic technologies can also be used to identify key biomarkers such as genes, proteins, and metabolites during specific food spoilage or pathogenic activity. These biomarkers can subsequently be used in formulation of rapid detection methods for food spoilage and pathogens. Strategically, these molecules can be used to identify molecules or physiochemical parameters that are critical for the growth of such microorganisms, that can again have implications in the development of conditions that can adversely affect the growth of these organisms, that can again be used for its control. The fourth use of omic science may be in the efficacy testing of specific food molecules. Rapid efficacy testing of bioactive food components depends exclusively on availability of reliable biomarkers. Upon the administration of bioactive compounds, changes in gene expression or proteome level can be monitored accordingly, both again in vitro or in vivo conditions or models, to identify a reliable, suitable biomarker. Hence, based on the level of change biomarker, the efficacy of the food component can be established even without understanding the exact mechanism of the change taking place. The fifth use of omics science may be in the usage of genotyping. Omic technologies can also be used to check the response of genetic polymorphisms upon certain nutritional components. Subsequently, this can be used to check the differential responses to nutritional components that can be used again to identify polymorphisms in a population. This may help in identifying genetic predisposition of an individual to specific diet-related disorders. The sixth use of omics science and food science may be in evaluation of quality of manufactured, processed, or even the raw foods. Comparative analysis based on proteomic or metabolomic profile of foods from different geographic origin can be done. Now this can help to study either the distribution of different foods over a geographic area or again identify its specific quality. These data can also help to identify the genetic predisposition to specific diet-related disorders or the distribution of diet-related diseases. The seventh use may be the development of knowledge-based functional foods. The expression profiles of specific bioactive components along with knowledge of the molecular pathogenesis can be used appropriately for designing novel functional foods. By using a combination of this nutrient mixture, it would be possible to prevent specific diseases or again development of personalized diet for disease person. Some more use of this science in food science may be production of food ingredients. Applied genomic technologies can also be applied to increase the yield of many food ingredients such as vitamins, amino acids, carbohydrates, yeast extracts, and so on from microorganisms or plants. Metabolomics can be used to identify key metabolites that control the product formation, and hence this information can be manipulated by metabolically engineering the microorganisms to increase the flux and the overall yield of the product. Another application of omics science is in the field of food allergy. Omics and food allergy. Food allergy is another area in which applied genomics will prove in particular to be very essential at various levels. Food allergy, mainly caused by certain proteins in our food, is now an increasingly important issue for the food industry. It is based upon the interaction of the host immune system with specific low digestible proteins, often with specific post-translational modifications. Even with the advancement of the technology, very little is known about the mechanisms that play a crucial role in the development of food allergy. Hence, in order to improve our understanding of the whole sensitization process leading to food allergy, immunologically oriented mechanistic research in combination with the new applied genomic technologies as a part of an omics science will therefore result in an increased understanding about this complex process or the problem of food allergy, which might ultimately help in eliminating the problem therein. Coming to the conclusion part, dear students, today we learned about the various concepts of omic technology and its application in nutritional science. We now know that omic science is a various combination of genomics, the proteomics, the transcriptomics, and again the metabolomics. Each of this is a separate field which is having diverse application in different fields, However, in nutrition science, the same can be applied in different combination in different applications for either the development of novel products or again personalized diet, including others. Thank you.